everybody. So I am going to use this video to kind of walk you through how to work some monohybrid problems, okay? So the first thing that we have to do is know how to set up a Punnett square. So what we do is we take the gametes from one parent and we put them across the top. We take the gametes from another parent and we put them on the side. So I'm going to label this one gametes A. I'm going to label this one gametes B. So you could say that, up here gametes A are the gametes from the mom, and then gametes B are from the dad, okay? And then in the Punnett square go the possible outcomes for the offspring, okay? So here in this box are going to be all of the possible genotypes of our offspring. Let's look at some other things that we need to know in order to fill out a Punnett square. So first of all, when we're setting up a cross, we use letters to represent the genes of an organism. And you saw that in both the dog called Spot activity and the Beaker babies activity. Well, how do we determine what letter to use? Usually, it is the first letter of the dominant allele, okay? Not all the time. Sometimes like you saw in the dog called Spot activity, it's really hard to tell the difference between a capital P and a lowercase p. So sometimes it might be beneficial to use the first letter of the trait, okay? But most of the time, we're going to use the first letter of the dominant allele. What kind of letter is used for a dominant allele? We saw in the Nearpod and in Beaker babies and dog called Spot that we use a capital letter. And for a recessive trait, we use a lowercase letter to show us that that is recessive. So remember, a dominant allele is going to cover up a recessive allele. Let's do just a little bit of practice. In a certain plant, the gene for red flowers, big R, is dominant to the gene for white flowers. So right now we're just going to practice figuring out genotypes and phenotypes. So what is the genotype of a heterozygous plant? Now remember, genotype means genes. So what I'm asking you for here is the actual genes of this organism. So heterozygous, we learned that hetero means different. That means that a heterozygous plant is going to have different alleles. So big R, little r. What about the genotype of a homozygous red flowered plant? Okay, so again, if you look back up here, we saw that red was dominant, and we know that homozygous means same. That means that we're going to have two dominant alleles, big R, big R. The genotype for a white flowered plant, we know that white is recessive. And again, the only way for a recessive trait to show is if we are homozygous for that recessive trait. So we know that a white flowered plant would have to be homozygous recessive, little r, little r. D and E ask you about phenotype. So what is the phenotype or physical characteristics, in this case, the color of a plant with two dominant genes? Okay, remember, dominant always covers up recessive. The dominant color is red. And then letter E, what is the phenotype of a plant with one dominant and one recessive? Again, because it has that one dominant allele, it's going to be red. Okay, I'll do two practices with you, and then the rest of this is on your own, okay? So what if we cross a homozygous red flowered plant with a homozygous white flowered plant? Homozygous red is big R, big R. Homozygous white is little r, little r. So again, one parent is going to go across the top of my Punnett square. The other parent is going to go here on the left. So I'm going to put my first parent, big R, big R. I'm going to put my second parent here on the left, small r, small r. Make sure you keep the parents together, okay? One parent goes across the top, the other goes across the side. I sometimes get people that try to divide them up, and it will mess up your results. So now to do the cross, all I do is say, okay, here's a big R, here's a small r. I'm just combining them in a grid, right? So big R, small r, big R, small r, big R, small r. Again, the dominant allele, we're going to write that first. It makes sense. If someone is dominant, they want to go first, okay? And big R, small r. So now let's talk about how to find the genotypic and phenotypic ratios. So remember, if you haven't already written this into your notes, you should, because you need to understand how to calculate this ratio. So go back to my Nearpod where I talk about how to calculate this if you need to. So remember, genotypic ratio is the number that are homozygous dominant compared to the number that are heterozygous compared to the number that are homozygous recessive. So how many do I have here in this Punnett square that are homozygous dominant? And I'm only talking about the offspring here, not the parents. I don't have any. So I'm just going to write a 0. My next value needs to be the number that are heterozygous. So I've got 1, 2, 3, 4. And then finally, I want to find the number that are homozygous recessive. So not here, or here, or here. So that value is also 0. 0 to 4 to 0 is my genotypic ratio. Phenotypic ratio, you'll remember, is the number of phenotypes. And so in this case, we have the number that are going to be red compared to the number that are white. It's the number that show the dominant trait to the number that show the recessive trait. So how many red or dominant flowers am I going to have? 1, 2, 3, 4. And how many recessive am I going to have? None. So my phenotypic ratio is 4 to 0. Okay, the last thing you need to know how to do is calculate probability. What is the probability of having a heterozygous offspring? Remember that probability is the number of times an event happens divided by the number of times it could happen. So the probability of having heterozygous offspring, we have 1, 2, 3, 4 out of 4, okay? So that is 100%. What is the probability of having offspring that express the dominant trait? So red, 1, 2, 3, 4. Again, our probability is 100%. Okay? All right, let's do another example. This one is going to deal with plants. Mendel found that the allele for tallness in plants, big T, is dominant to the allele for shortness, little t. What would be the possible results if a heterozygous plant self-fertilizes? So all that means is our heterozygous plant reproduces with itself. So heterozygous, again, is different alleles. So big T, little t. And since it's the same parent, big T, little t. So now I want to do my Punnett square. Here I have big T, big T. Here I have big T, little t. Here I have another big T, little t. And then finally I have little t, little t. Okay? So what I did here is just simply asked you to write out what the cross is, what the parents are. Okay? So that's big T, little t, crossed with big T, little t. Okay? Let's find our genotypic ratio. Remember, it's the number that are homozygous dominant, the number that are heterozygous, and the number that are homozygous recessive. So how many are homozygous dominant? 1. How many are heterozygous? 1, 2. And how many are homozygous recessive? Just this one. So our genotypic ratio is 1 to 2 to 1. Now I'm spacing it out, but that's really just so you can see everything. You don't necessarily have to space it out on yours. What about the phenotypic ratio? How many of these plants will be dominant? The dominant trait is tall or big T. So this one's going to be tall. This one will be tall because it has at least one dominant allele, and this one will be tall. So that's a total of 3 dominant to 1 recessive. 3 tall, 1 short. Let's do our probabilities. What is the probability of having a homozygous tall plant? Homozygous means same allele. So here we have our homozygous tall plant. 1 out of 4 possible outcomes. So you can write this a couple of ways. You can write it as a fraction, 1 out of 4, or you can write it as a percentage. 1 out of 4 is 25%, or you can write it as a decimal, 0.25. Okay. So now the last question here, when a plant self-fertilizes, is there still genetic variation between the offspring? Well, we see here that our parents are heterozygous, and we have possible outcomes that are not heterozygous. So yes, there absolutely is a chance for genetic diversity, even if the plant is self-fertilizing. All right. So I'm going to do one more with you, and that's slide 10 down here. It says 2 fruit flies with normal wings are crossed. Among their offspring are 89 with normal wings and 33 with dumpy wings. So I put a Punnett square here in case you want to work through it. Okay. I'm actually not going to do this whole one with you. I just kind of want you thinking about, what do you think the dominant trait is? What do you think the recessive trait is? And then how is it possible for these normal flies to have offspring that are different from them? Okay. So we will have a chance to work through this in class. I hope that this was informative for you, and I hope you enjoy. People really do kind of like doing these Punnett squares once they get the hang of it. Bye, guys.